This video is an overview of fermentation. So we're going to look at alcohol fermentation and we'll look at lactic acid fermentation very briefly. But before you can learn about that, you need to make sure you understand glycolysis. So make sure you were in class, read your notes, or watch the previous video on glycolysis before you watch this one. What we do know is glucose is converted into pyruvate through the process of glycolysis. Glycolysis takes place in the cytoplasm or the cytosol of the cell. And obviously, we don't see all 10 steps here, but what we do see are the energy outputs that we care about. So for every 1 glucose, there's a net of 2 ATP generated, and that ATP can be used by the cell to do cell work. We also see the reduced electron carrier, NADH, and that should ring a bell from when we learned about the light reaction and we saw NADPH. So this is similar. NADH is carrying the electrons that were stripped off of the glucose or the G3P in glycolysis. Now, if oxygen is present, usually pyruvate will go to the mitochondria. And we haven't talked about what takes place in the mitochondria, but later on we will talk about how pyruvate will then be fully oxidized. It will really be stripped apart for its energy. And also NADH will go to the mitochondria, and it will do something that is similar to what we saw in the light reaction, where we saw chemiosmosis, and we'll talk more about that later. If the NADH goes to the mitochondria when oxygen is present, it will drop off its electrons and it will be oxidized to NAD+, and that NAD+ can come back and be used in glycolysis. Now, if there's no oxygen and NADH is not going to the mitochondria, that means we have a very small pool or limited pool of NAD+. So if we don't have NAD+, then glycolysis cannot continue. And if glycolysis cannot continue because there's a lack of NAD+, that means we stop making ATP for the cell. Even though 2 ATP is very little, in some cells, that's actually considered a lot if you're talking about small single-celled organisms. So there's a mechanism to kind of get around that to allow glycolysis to continue even without oxygen. What the cell can do is it can convert pyruvate into this molecule, acetaldehyde. And when it does that, it makes carbon dioxide. So if you think about beer and champagne and bread, where do those bubbles come from? This is where they come from. So we have the bubbles in beer and champagne, and the bread rising, that gas that's produced that allows it to rise is this CO2. Then the cell has this enzyme here called alcohol dehydrogenase, and we're going to say it's ADH1, and that will convert acetaldehyde into ethanol. Now, that is not the goal. The goal here is not to make ethanol, but to instead make NAD+. So what's happening is the acetaldehyde is actually reduced. So we see it's gained some hydrogens here and here, then they weren't over here and here before. And at the same time, our NADH is oxidized into NAD+. So we've removed the hydrogens and the electrons off of NAD, and we've put them onto the acetaldehyde to make ethanol. Now we have NAD+, and glycolysis can continue to make small amounts of ATP. Organisms that do this, so we have bacteria will do this, some alcohol fermentation, and fungus are the ones we really know, so yeast. So whenever we're talking about any alcoholic beverage, it's usually yeast doing that fermentation, making the alcohol. We see something similar with lactic acid fermentation. And in lactic acid fermentation, we have bacteria and we have animals like us. So we are humans, are animals. We also do lactic acid fermentation when there's no oxygen. So bacteria might choose to do lactic acid fermentation even in the presence of oxygen. It depends on the species. Humans, animals, we are only going to choose this as a last resort because it doesn't make a lot of ATP. So only when you've exerted yourself and you have lower levels of oxygen, it doesn't actually need to be that much lower, but if you have lower levels of oxygen, your body will start to resort to using some lactic acid fermentation, with the same rationale as the alcohol fermentation. So we see here the glucose gets converted into pyruvate. Just like before, we made a net of 2 ATP and we made 2 NADH electron carriers. If NADH is not going to the mitochondria because there's lower levels of oxygen, we need a way of replenishing our NAD+ in a different way. So the pyruvate will be converted into lactate. And there's an enzyme there that does that process. You don't need to know the name of it. But again, that enzyme there, it is reducing the pyruvate while it's simultaneously oxidizing the NADH. This is what we care about, making NAD+. So we do this in our muscles. Bacteria will do this and they produce foods like yogurt and kimchi and sauerkraut and pickles. That tangy taste of those foods comes from this lactic acid fermentation. And I hope this is helpful.